Hi, and welcome to the first session of Spatial Snippets. I'm Sarah Church. I am a senior scientist in the Translational Science Department at NanoString, and today I'll be talking about the GeoMx Canine Cancer Atlas. There are many challenges in oncology research, such as including that cancer is a very diverse disease. It's not just one disease, there are many, over hundreds of types of cancer. Also, there's a large amount of heterogeneity even within each of those individual diseases within the tumor microenvironment. And most of all, not all patients respond to treatments. And it really leads to the necessity to have biomarkers and signatures to understand who responds and who doesn't respond to particular treatments, so that people aren't getting expensive treatments or having adverse side effects to treatments that could be avoided. So there are many preclinical models that have positive and negative roles within oncology, but it's well recognized that there are many problems with mouse models. Particularly in that the efficacy in humans translated from mouse is very low. So one of the main reasons, or the main reason why clinical trials fail, is that the model that was in the mirroring model was not translated into the humans. And this is widely recognized by the National Academy of Sciences. And one of the main reasons is that mice don't naturally develop cancer, so they're all artificial systems, and that rodent physiology is quite different than human physiology. As a result, in more recent years, they've skipped preclinical models and really done the first rounds of testing of combination therapies and new therapies in humans themselves, which is quite was not cost effective and has many high failure rates. So what are the advantages of canine comparative models? Pet dogs develop many spontaneous diseases, including cancer, and they just demonstrate comparable genetics, molecular, and clinical features with those found in humans. The incidence of cancer in pet dogs is also 2.5 to 5 times higher than humans. And also, they have unique and very similar immune systems to humans, so it's a unique opportunity for us to study immunotherapies that are going into humans. So there are several resources that exist to support clinical trial work for pet dogs. They're listed here for any of those that are interested. So NanoString has found that it's really important to create tools to be able to characterize canine tissues in all types of health and disease, not just oncology, but to veterinary medicine as well as cancer and aging programs. As part of this, we developed the GeoMx Canine Cancer Atlas, which is really looking at tumor and immune response of canine tissues to therapy and to just overall biology. This was a panel that was developed by a consortium of different canine researchers. It has expansive coverage of 1,962 genes as well as controls, and those cover over 110 different pathways. It's meant to be a pan-tumor panel, so it covers the most common types of biology that are specific to tumors that are common in canines. It's very sensitive. We've can detect over 1,000 different unique genes. And it also has cross-platform applications with bulk profiling through our nCounter Canine IO panel, which has overlapping content, so you can compare spatial and bulk gene expression analysis. You also are able to customize the assay with up to 400 different genes of interest. And all of that is read out with the GeoMx next generation sequencing output. So here's just a list of the pathways that are covered by the GeoMx CCA assay. On a high level, that includes adaptive immunity, cell function, immune response, innate immunity, metabolism, physiology and disease, and signaling pathways. We've already started profiling different types of tissue morphology, also tumor microenvironment, and immune-infiltrated hotspots on a number of different tissues, both normal and cancerous tissues. So here on the left, you have an example of 6 different tissues that we've profiled using our morphology markers from GeoMx. And then on the right, you can see that based on that profiling, you get to measure gene expression in that spatial way based on canine-specific tissue heterogeneity. So here's just a differential expression of looking at hemangiosarcoma and astrocytoma and what genes were coming up based on those 2 different types in different areas of that cancer tumor microenvironment. The GeoMx Canine Cancer Atlas assay is very easily put into a data analysis pipeline. You're able to integrate all kinds of other spatial analysis, such as H&E or immunohistochemistry. You can also integrate digital pathology. You can integrate bulk expression, as we talked about with the nCounter canine panel. And then you can also incorporate any clinical data. This makes finding new biomarkers and new signatures easy to do and already built in to the GeoMx instrument and processes. So thank you for joining me for this spatial snippet, and until next time.